Good morning, Prof. Andre. Thank you for allowing me to meet you to talk a little about genetic testing. Please introduce yourself.
Thanks a lot, Anton. Yeah, I'm Andre van Niekerk. I'm a Paediatrician and a Paediatric Pulmonologist by training. And working in the field as a Pulmonologist, I had to deal a lot with children with recurrent infections. And that's where my interest in primary immune deficiency diseases started. And lately, I've been appointed as the chair of a new chair that was founded at the University of Pretoria, and that's the chair for inborn errors of immunity and allergology.
Okay. Tell me now, for us that's new to the term inborn errors of immunity. What is inborn errors of immunity?
So, if your immune system is being naughty, it does so in different ways. It doesn't just behave in one way. I always say to the students that a naughty immune system may decide to fight things that it should not fight. And that's allergy. So if our immune system starts fighting innocent things like the cat or pollen, that's an error of immunity, and that's what we call allergy. Or the immune system may, for example, not fight germs when it's supposed to fight germs. And that's called primary immune deficiency if it's got a genetic origin. Then we call it the primary immune deficiency. Or the immune system may decide to fight your own body tissue. And that's called autoimmunity. Or the immune system may decide to overreact. And that's called autoinflammation. Or the immune system may decide not to fight cancer when it should be fighting cancer cells. So all these things go together under one umbrella, and that's illness or misbehavior of the immune system, and that's why we call it inborn errors of immunity if there's a genetic background to the misbehavior of the immune system. In other words, primary immune deficiency is part of inborn errors of immunity. And many patients who present with a primary immune deficiency will also suffer from allergy or autoimmunity or run the risk of developing a cancer or a malignancy at some stage.
I've never heard it explained in that fashion. Thank you. That really helps. You touched on genetics.
Yeah.
You are also responsible for genetic testing.
Yeah.
How does one get access to genetic testing?
Well, I think we must first of all just understand why would genetic testing be valuable. Why do we want genetic testing? Now, as I said, we're dealing with inborn errors. In other words, there's a genetic background behind the problem. And that's why we would like genetic testing in all our patients, to understand what the genetic background is. And we also need to understand what a gene is and what a gene does. So we've got lots of genes, and every gene is the program to a protein, and every protein that your cells produce has a function. And it could be a structural function to form part of your body, or it could be a function that that protein must execute. And the immune system relies heavily on these proteins. These proteins coming from our genes, they perform many functions in the immune system. They are the signals to things. They are the signals to the development of cells. They are the communication signals between the different cells of the immune system. And I can go on and on and on and on about what all these different proteins do as part of our immune system. So if one of those genes are wrong, in other words, we are now encoding for a faulty protein, and if that protein plays a crucial part in the immune system or an important part in the immune system, that will then lead to some sort of immune deficit. And remember that that deficit can be a loss of function, or it can sometimes also be a gain of function that we don't want, like in autoinflammatory conditions, for example. So why do we want to study genes? We want to study genes because they are behind the problem. That's why we want to study them. And in many of the immune PIDs, and we now know of about 485, I think that's the last list that we had, there are now 485 genes that are specifically linked to a primary immune deficiency disease. And in other words, many of the PIDs are what we call monogenetic deficiencies. There's one gene, and because of that one gene that's got an error, we've got a PID that can be linked to that one gene. But what makes it difficult is that an abnormality in one single gene could present in different clinical ways. In other words, all patients with that same gene abnormality don't necessarily look the same in the bed. So we often sit with patients with clinical pictures that we are finding difficult to explain. And to explain what's going on, that's why our genetic testing can help us a lot. So the results of a genetic test helps us understand the mechanism of a specific disease, and at the same time it gives us an idea of what to expect in future. In other words, it could give us an idea of what may happen to this patient or what may not happen. This patient may be more prone to a cancer later, and therefore we want to screen better for cancer in the future. That's an example. And it also, with all the new technology that's developing, gives us an idea of what the chances are of actually fixing that gene. That's the problem. But I must at the same time just remind you that there are also many PIDs that are not monogenetic, where it's more than one gene that plays a role. Or there are PIDs at this stage where we don't understand the genes behind it yet. And therefore, genetic testing often gives us very valuable information, but sometimes we still don't understand after we've seen the results.
Tell me, Professor, how is genetic testing done?
So we need some of your genes. So the easiest for us is just the blood sample. Get the blood sample and isolate your DNA from the blood sample, and that is then sent to a lab, where the lab uses very fancy technology. There are different types of sequences, and the one that's most commonly used at the moment is called next generation sequencing, and the DNA is literally read by these fancy machines or sequences, and the data is collected. And then comes the problem. The data must be analyzed. Because if we read your genes, we collect a massive lot of data. So we really need people who are skilled. We call them bioinformaticians, skilled people to analyze the data and to then give us a result in terms of your genetic testing.
Can genetic testing be done in South Africa?
Yes, we can. There are different ways of doing genetic testing. So we basically got three processes. The one option is to do what we call targeted panel testing. So targeted panel testing, you've got about 25,000 active genes. So targeted panel testing is where we say to the lab, we suspect that you've got a PID, and they will then only run a selected panel of genes. We guide them in terms of the panel of genes that we would like them to read for us. At the moment, for example, one of the targeted panels that we do read about 500 of the genes, and they will read those genes and only give us an answer on those genes. If we don't get an answer there, the next step would then be to do what we call whole exome sequencing. So exome sequencing is where we read all your active genes. But remember that your genome contains far more pieces of information than just the active genes that are being transcribed, and that's called the genome. So if whole genome sequencing, first of all, exome sequencing is where we just read the active genes, and we could then go a step forward, a last step, and that's to read your entire genome, and that's whole genome sequencing. So not all of these tests are done routinely in commercial labs in South Africa, but we've got networks all over the world that the tests are being sent to. So there are units specializing in the targeted panels, There are units where we could send whole exome sequencing to, and we can even send off whole genome sequencing. All of it is not done in South Africa. Some of it can be done in South Africa, depending on what we're looking for. And unfortunately, at the moment, some of the cheapest and most cost-effective results come from outside of South Africa. And we often then send these samples off to places like America or the UK or Europe. And the problem there is then funding, because our medical aids are refusing to fund tests that we send off outside of the border of South Africa often.
So it can end up to be a costly exercise for the patient.
So for PID specifically, the targeted panel that we do at the moment is $349. That's the price. And of course we need a courier, a lab company to isolate your DNA from the blood sample and send it off for us. And they charge us for a consultation fee. There's always genetic counseling that goes with genetic testing. For a consultation fee, together with a courier, it's about 1,300 Rand extra. So roundabout, depending on the exchange rate, we're looking at 7,000-8,000 Rand for a targeted PID panel. So I don't think that's that expensive if you compare it to all the routine functional tests that we are doing, and the quantitative and qualitative, just measuring your antibodies and counting your cells. That's also expensive. So I don't think genetic testing is that expensive for the targeted panels. When we're doing whole exome sequencing, it becomes more pricey. At the moment, a whole exome is about, I think it's $2,500. I'm not sure of the price of whole genome, but it becomes more expensive. The more info you want, the more you pay for it. No, but you know what? This is technology that's becoming much cheaper. 3 years back, we paid for a targeted panel 18,000 Rand. And it's now down, and that only included 99 genes. We're now sitting at something like 472 genes, and it's costing us 7,000 Rand. So the technology is becoming more available, it's becoming cheaper. It's the science of the future, because we're going to deal with precision medicine.
So if I understand it correctly, then if there's no specific need for a patient to do gene therapy for ensuring quality of life, you might as well postpone it a little, because it will be more cost effective in a year or two.
Well, that's a difficult question. I think what we would like is to actually, on every one of our PID patients, have access to genetic testing. And as I say, it often gives us answers that we don't fully understand. Many of these diseases overlap. They present clinically when you stand at the bed of the patient. It could be one of many possible diseases. And what we then do, or possible genetic defects. And what we then do is we do repeated functional or quantitative blood tests. We measure your immunoglobulins repeatedly. We measure your blood cells repeatedly. We count them repeatedly. We look at their function repeatedly, and then look at the pattern. And based on the pattern of what we see in combination with the history that you've given us and what we see clinically when you examine you, we sort of form an idea that this should be the underlying genetic problem. But that's not an accurate science, because many of these diseases present in a similar way. And it makes it much more accurate if we could have a genetic answer. So my answer is if I could have my way, I would do at least a targeted panel on almost every patient that I see. There are cases where it's not necessary, but at this stage it's not possible. In certain cases where we need to take critical decisions, urgent decisions, like for bone marrow transplant, for example, or stem cell transplant, we would really like a genetic test in order to take that decision, because it really forms the foundation of an accurate decision to have a genetic test. So in all our patients, we discuss it on an individual basis. I say to parents, in your case, I would really like a genetic test. Can't we make a plan? In your case, I'm not that worried, and therefore we could postpone it or never do it maybe. But I hope we're going to get to the day where this technology is so readily available and cheap that we could offer it as a first-line investigation.
Prof, you mentioned counseling.
Yeah.
What's the need for counseling?
So, first of all, genetic testing is difficult to the doctor. It's difficult for patients to understand. There's money involved, and it's got implications. And therefore, no patient should be submitted to genetic testing without proper information. And proper information going both ways, going towards the patient and the doctor. And we've got people who are specially trained as genetic counselors to sit with the patient and discuss what the test could do for you, what it can't do for you, what the outcome could be, because with every genetic test that we do, we may find an exact answer. We will also learn what's not wrong with you, but we may not learn what exactly is wrong with you, depending on the panel. If your panel, for example, didn't include the genes that's behind your problem, or the gene that's behind your problem, we're not going to find an answer on that panel test. But at least the panel will tell you what's not wrong with you, and that may be important to the doctor. And because genetic testing is an involving science, very often we get what we call VUSs, or variants of unknown significance. So these results will come back, and it will flag genetic variations in your genes, and we won't know what it really means because we don't have enough information. And patients are often insecure about, do they want to know these things? Don't they want to know these things? What will it mean for the future? Do I want to know what's going to happen to me in future? So there's a lot of information around genetic counseling, and I don't think any patient should do a genetic test without genetic counseling.
Thank you. Tell me, on identifying the problem genes, is it possible to remove them, change them, alter them, fix them?
Well, that's what we ultimately hope to be able to do one day, because behind the problem sits a problematic gene. And we would like to be able to fix that gene. Because if we could fix the gene, then theoretically we could fix your problem. And you could have a total normal life if we could fix that gene. Now, fixing genes can be done by replacing them, and that's what happens with stem cell transplant, is by replacing them. Or, and as you know, stem cell transplant needs a compatible donor. And that's the big problem with stem cell transplant, is to find a match, and preferably a perfect match. Because if the match is not great or not perfect, you sit with all post-transplant sort of problems. The graft versus host reaction, rejection, etc., etc. So we would like a match, and that's a problem with stem cell transplant, is finding a match. Now, if we could fix the gene in another way than replacing it, it would be far more helpful. And that's where the whole world is going in terms of research. And that means that we would like to, if possible, add a gene. If there's a faulty gene, add a gene to your stem cells. Or we would like to edit a gene. So if you've got a faulty gene and we know what the fault is in your stem cell, is to use technology to literally go and fix that gene or edit that gene. And that's called genetic transfer or genetic editing. An highly advanced technology that's not commercially available at this stage. But that's the future of precision medicine.
To identify a problem beforehand, it would then be advised to screen a baby as early as possible.
It's a big topic. There are many ways of screening. If you know which gene is the problem in a family, then that fetus before birth could be tested to see if the baby has the same defect. And one will do that if you would consider a serious situation like a termination of pregnancy, for example. That's legal in South Africa to do that. But then you need to know which gene it is that you specifically want to test for. Another way of screening is after birth on a simple, just a piece of paper, a special paper with a spot of dry blood on it from the baby. We could do basic tests to see whether T cells are maturing or B cells are maturing. They are called CRECs and TRECs. And this is the most common form of screening that's done in the world. Because that's, I think it's less than 1,000 Rand for that test. And it's done on a dry blood spot. And it could warn us in advance that this baby is at risk of a severe immune deficiency, like severe combined immune deficiency SCID, for example. And if we know that in time, we could immediately cocoon that patient and take good care of that patient to prepare that patient if it's going to end up in a stem cell transplant, for example. So that's a way of screening, is at birth. And then patients who present with suspected immune deficiency, we've got first-line testing that we do in the lab, basic screening tests, just the blood count, just count their immunoglobulins and see how they responded to vaccines. That's also a way of screening to see whether we've got an underlying immune deficiency or not. So there are different ways of screening. It's a big topic, but something we need to embrace.
Newborn screening. The most accurate way would be after birth.
So newborn screening is what I refer to as the dry blood, is the spot test, and that's where we just measure TREC and CREC. That's a bit technical, but TREC and CREC is a molecule that we measure in the blood, and those molecules are protein molecules. They are absent if the patient doesn't have maturing T cells and B cells, and remember those are the brains of the immune system. So if they're absent, we can test it, pick it up on a simple test like that. But if you mean screening, we're going to screen the patient's entire genome. It's impossible. We can't use whole genome screening or exome screening or even targeted panels at birth because that's just too expensive to do it on every baby. It's not feasible. But if there's a very strong family history, and we're very worried that this baby may have a specific gene that's abnormal, of course we could use that then at birth. But for general population screening at this stage, it's basically just measuring the TRECs and the CRECs, and they will pick up the important patients at birth and warn us these patients must be managed differently.
Meaning that as a fetus, there's not much testing that can be done.
There can be tests done, but then you need to know what to test. So if there's a strong family history and we're really worried that this baby is going to be born with a specific genetic defect, then there are tests that can be done before birth to identify if this baby is actually at risk. And if the baby is at risk, it will actually diagnose the baby before birth. And if the baby is then diagnosed through genetic testing, the parents could then decide on termination of pregnancy if they want to or if they don't want to. So the purpose of antenatal screening is actually testing. We shouldn't actually call it screening. We should call it testing. Because you expect a certain problem, you test for that specific problem, and if it's present, you take a decision on it. But screening as such is what's done at birth on dry blood spots.
Are there funders that can actually fund this? Would medical aids pay for it? How does one go about getting funding for newborn screening in general, or in specific cases where there's families that have a possibility of carrying the gene forward?
So funding in state patients for generalized newborn screening is not available. It's not an option at this stage. It's not being done. Any person can buy a TREC and a CREC screen for their baby, and you must get it funded. And as far as I know, it's around about 1,000 Rand. So that newborn screening is available in South Africa. One of the private labs is doing it. Impact Lab offers the service for TREC and CREC screening. but you need to request it specifically. It must be done. You can do it at any time, but immediately after birth is the best time, and you must supply the funds to pay for that. And anybody in the country has got access as long as you pay for it. But it's not government subsidized, and I'm not aware of medical aids paying for newborn screening in terms of Trek and Crick, out of their risk benefit in any case. So that's in terms of the Trek and Crick screening. For genetic testing, whether we're doing a targeted panel or a whole exome, or we're doing a whole genome test, you can choose what you want to do, but at this stage, funding is really a problem. Even patients on medical aids, most of the medical aids are refusing to pay these tests. I think it's something that should be lobbied about. These tests should be paid. They are valuable tests. They are international standard of care now, and therefore we should lobby medical aids to pay these tests. They pay many other tests, and they should be paying these genetic tests as well. Of course, we should approach it in a logic way. We can't expect a medical aid to just do a whole genome funding on every patient. But at least targeted panel testing, I think, is something that should be lobbied for, and that we should try and get that funded through medical aids. Any patient where the medical aid doesn't fund it, it's readily available. We can just pay it. There are research projects going on in the country where genetic testing is funded by a sponsor that's involved in research. So that is another way of maybe getting a genetic test, is to become part of a research project. And I must say we're currently grateful that an international foundation, Jeffrey Medell Foundation, they are involved in supporting on a research basis. And we do get some access there, but it is not free for all.
Prof, thank you. So it is available. You just have to pay for it currently from your own pocket.
Yes, anybody can buy it. It's freely available, but you have to get money to do it.
Prof, thank you. I think you've answered a lot of questions and a lot of clarity for someone that is considering gene therapy. So from my side and PiNSA side, thank you for the time. Thank you for the explanation. And hopefully we can lobby it in the correct way in future. But thank you.
Pleasure, Arthur.
So